Hello friends, this is Chetna. Today we are going to learn about genetic engineering. We'll see a brief overview of genetic engineering. So let's start. Drawing heavily from cellular, molecular and developmental biology, biochemistry, engineering technology, medicine and other modern sciences. The discipline of biotechnology was formed to develop various applied concepts like cell fusion technology, bioprocess technology, structure based molecular designing, then cell programming, and many more. Now, collectively referred to as genetic engineering, or which is also known as bioengineering. So, biotechnology can also be defined as the integration of natural sciences in order to achieve the application of organisms, cells, parts thereof, and molecular analogues for products and services, which comes under European Federation of Biotechnology, also known as EFB. It's a General Assembly formed in 1989. So, brewing wines, making cheese, baking breads, manufacturing liquid oral drug formulations by special fermentation processes. Even septic tanks of Mohenjo-daro and Harappa tell us that biotechnology was known to man, albeit in a crude form in the ancient times as back as 6000 BC. But it is only in the late 18th century that the understanding of the underlying microbial process started taking shape through the work of Louis Pasteur, Madame Curie, and other scientists all over the world. Development of early vaccine, for example, smallpox vaccine, from animal sources was a step in that direction. But the real breakthrough came in the form of the discovery of the double helix, that is DNA molecule, by Nobel laureate Dr. James D. Watson and Dr. Francis Crick in the mid 50s, which is also known as Watson and Crick model. So while cloning, this cloning part we will see in the next presentation. While cloning gives only one facet of the tremendous opportunities and avenues thrown open by genetic engineering. The revolution unshared in by it is already covering disease prevention and treatment. Then pharmaceuticals, neurobiology, agriculture, food processing, and many more fields. The human body is a complex biochemical factory where thousands of chemical reaction, also known as processes, are going on continuously inside our body. These reactions are catalyzed by proteins made from genes. The state of human body, healthy or otherwise, is determined by properly coordinated functioning of these genes and proteins. So tremendous advances in molecular biology have enhanced the understanding the genes and the structure of many of their proteins, which ensures healthy life. In turn, this knowledge has made it possible to isolate these genes and to produce those rare but valuable variety of biological molecules, which are also known as proteins molecules in a large quantities. So far, only a limited number of proteins useful for prophylaxis, diagnosis, and therapy or treatment in human beings have been made available, and genes for these proteins have been cloned and expressed. The best example is human insulin, the first recombinant protein to be commercialized. Previously, insulin was extracted from slaughtered animals. The most important aspect of these products are that they are pure, much safer, abundantly available, and comparatively cheaper. So we will see some categories of the genetically engineered proteins, or we can also call it as a products. Genetically engineered products being used today are four types. First, vaccines, second, substitute, third, treatment, and fourth one is enzymes. So, in vaccines for hepatitis, herpes, malaria, etc., then substitutes to correct the deficiency of an otherwise normally available product in the body due to genetic defects, which is also known as substitution therapy, such as insulin in diabetes, growth hormones, dwarfism, and clotting factors. For example, tissue plasminogen activator, which is also known as TPA, which is used in hemophilia. Some of these are used in therapy also. So, third one is a treatment, which is also known as therapy. This therapy of diseases, that is TPA, interferons in the viral infections, cancers, then calcitonin in the osteoporosis, then interleukins in the cancer, etc. And the fourth one is enzymes, the biological catalyst that facilitate biochemical reactions going on 24 hours in the body. Without them, life sustaining metabolic activities would come to a standstill. So, today, science does not know everything about genes. In human genome research across the globe, so far only 50,000 genes have been identified and 20,000 express sequences have been mapped, while 20 billion pairs are yet to be mapped. The complete sequence has 3 into 109 base pairs, that is 109 base pairs. That is magnitude of work on hand. This all data is a tentative, and day by day it will change. So genetic disorders will continue to show their presence in all the time to come. Genetic origins will be clearly established for more and more clinical conditions in the continued research. Increased medical and surgical interventions will further add to the diseases caused by polygenes. What are polygenes? These are products of genetic mutations and non-genetic factors interacting in a ways that are not clearly recognizable. Yet genetic engineering today holds a great promise to future generations to reduce morbidity. What is morbidity? It's a sickness and its prevalence. And mortality, that is a death rate, to increase the efficiency of human brain, to reduce, if possible totally eliminate birth of genetically unhealthy children, to lengthen the lifespan by slowing the process of aging, and to improve the overall quality of life. In all these processes, genetic engineering plays a very, very important role. So this we have seen a brief overview of genetic engineering, and also we have seen genetically engineered proteins in a brief. Thank you for watching.